Best way to find the lectures, did I tell you all about my YouTube channel?
Yeah, it's like Alex Smith.
Alan Smith Biology, yeah. So I try to upload them at the end of every day that I do the lecture. It may take a while, but it'll get up there. So a couple of you were asking me about the difference between an element and a compound and some rather basic chemistry. So I'm going to go over this fairly quickly, okay? An atom is the simplest chemical unit. Make sure you get that down. The simplest chemical unit is an atom. While there are particles that are smaller than that, they do not exhibit unique traits found in other substances. Is everybody clear on that? Things like protons, neutrons, and electrons. Protons, neutrons, and electrons. I've always found it easiest to make up a table. That helps me remember the difference between them. In column 1, I put particle. Column 2, I put charge. Column 3, mass. In other words, how much does it weigh?
Can you say the first three again?
I said particle, charge, and mass. Those are your 3 columns. The 3 types of particles: neutron, proton, electron. Neutron, proton, electron. For charge, put a 0 by neutron because they are neutral and have no charge. Put a plus by proton and a minus or negative symbol beside electron because they are negatively charged. For mass, neutron is a 1. Proton is a 1. And just keep things simple, we'll say that an electron is effectively got a mass of 0. Now you know there's no such thing as a particle that has no mass. But this would be like trying to take an elephant and a mosquito and compare their weight. The mosquito obviously has mass, but it would be so little compared to the elephant that it's not even worth mentioning. Is everybody okay with that? Makes it simpler. Now, if you look at a periodic table, which you will be allowed to use on the exam, by the way. So if you want to call one up on your tablet, phone, or whatever, feel free. The number at the top is the atomic number, That's the number of protons present in the nucleus. It's the number of protons. If you look below the symbol, you'll see a number that's probably a decimal point, about 4 or 5 decimal places. We are going to round that up to the nearest whole number just because it's easier. And that is called the atomic mass. The atomic mass.
Sorry, where is it again?
At the very bottom, right below the symbol. So if you look in the upper left-hand corner, you should see H for hydrogen, atomic number 1. Right below the H, there should be something like 1.0000. Do you see that? That's your atomic mass. And the way to tell the difference you want to know how many neutrons it has, you subtract the atomic number from the atomic mass. So if you take a look at element number 3, lithium, Li, second row, upper left-hand corner. Does everybody see that? So it has 3 protons, doesn't it? How many neutrons does it have? 4. How do I know that? Because the atomic mass is 6.96. We round that up to 7. Take away the 3, we're left with 4. Everybody cool with that? Electrons are the problem children. Generally, you should have as many electrons as you have protons. You should have as many electrons as you have protons. But as we're going to see, it doesn't always work out that way. Everybody with me? Yes, ma'am.
Can you repeat the formula again? Atomic mass minus number of protons?
Atomic mass minus atomic number.
Okay, atomic.
Since the atomic number is by definition the number of protons, if you take that away from the mass, it just leaves you with a whole number that tells you the number of neutrons.
Okay, thank you.
You're welcome. And don't worry, we're not going to go too deep into all of this. But from the volume of questions I was getting, it was kind of obvious that you all needed to get a little bit more background.
Today's lab, where would that be?
We'll talk about that at lab time.
I can.
It's all the way down on the left, just past the intersection. Not everybody's in the lab, believe it or not. We got some people who are taking the lecture, but not the lab. So, where was I?
Electrons and protons should be equal, but it doesn't always work out that way.
Okay, electrons. Electrons orbit the nucleus. They orbit the nucleus. And I'll just draw lithium for you. I said it had 3 protons, right? 4 neutrons. And all of those are located in the nucleus. They're packed together. And the rule is like charges repel each other. Like charges repel, they push apart. Opposites attract. If that's the case, then why are all those protons jam-packed into the nucleus? They're held together by something called the strong nuclear force. You ever heard of nuclear weapons or nuclear reactor? That is because they split the nucleus apart and release all of that energy. Put it into something you might see visually. Everybody sees the little silver part of my pencil. If that was pure uranium-235, it has as much energy as you would find in a metric ton of coal. Now you see why people are interested in it. A lot of bang for your buck. The electrons orbit the nucleus. They orbit the nucleus. And those orbits are set locations. They only can hold so many electrons. The first orbit is located closest to the nucleus. It holds a maximum of 2 electrons. It can never hold more than 2. Is everybody clear on that?
What is it closest to?
Somebody ask something?
What is it closest to?
The nucleus. The second orbit is bigger than the first. It can hold up to 8 electrons. Since lithium only has 3, 2 of them go into the first orbit, 1 into the second orbit. We're not going to go much beyond the first 3 orbits, the first 3 rows of the periodic table, okay? Does everybody understand that? An interesting quirk of atoms is that they want a full outer orbit. They want their outer orbit to be full. Now that atom that I drew up there, it only has 1 electron in the outer orbit, right? It's a lot of energy for it to go out and find 7 free electrons, isn't it? So it's actually going to use a trick. It's going to kick that electron out. The first orbit is now the outer orbit, isn't it? Is it full? It only has 2 slots. They're both full. End of story. We have just created an ion, I-O-N. An ion is a charged atom. An ion is a charged atom. If you take a look at the rows in the periodic table, you'll see a pattern. There are only 2 elements in the first row, right? There are only 2 slots in the first orbit. If you take a look at the second row, you'll see there are 8 elements. But there are only 8 slots in the second orbit. You see how this goes. Keep in mind, the atom wants a full outer orbit. It is going to do one of 3 things to get that. Number 1, it's going to do like lithium did and kick it out. Right next to that, we've got beryllium, don't we? Does everybody see that? Beryllium has an atomic number of 4, so it has 4 electrons. 2 of them go into the first orbit, 2 into the second. Is it going to be easier to grab 6 or kick out 2? Kick out 2. So that's going to have a +2 charge because now you have 4 protons but only 2 electrons. Does everybody see how this goes? If you jump that big gap and go all the way over to the big B, boron, I believe the atomic number of that is 5, am I right? Somebody looking at the periodic table?
Yeah, it's 5.
I'm such a nerd, I've got the periodic table memorized. This is scary.
Is that because it goes in order?
Huh?
It goes in order 1, 2, 3, 4, left to right.
Yeah. That one has 3 electrons in its outer orbit, 2 in the first orbit, right? 5 empty slots. It's going to kick out 3, so it's going to be +3. Does everybody see the pattern there? That's the first option. It can kick out electrons. The second option is it's going to share them. Carbon is a great example of this. Carbon should be right next to boron. It's element number 6. So it has 6 total electrons, 2 in the first orbit, 4 in the outer orbit. But it also has 4 empty slots, right? Rather than kicking things out or grabbing electrons, what carbon is going to do is it is going to share with another atom. It's going to share with another atom. And it will always form 4 bonds because it has 4 electrons. One next to it is oxygen. Nitrogen, sorry. Am I right? It's nitrogen?
Yeah. Yes.
Okay. Atomic number is 7. So 7 electrons, 2 in the first orbit, 5 in the outer orbit, right? It's only got 3 empty slots. Rather than trying to kick 5 out, it's going to grab 3. Does everybody see that? So it will form 3 bonds. And since we're adding electrons, we're going to form a negatively charged ion with a charge of -3. -3. Oxygen has 6 in the outer shell. It only needs 2, so it's going to form a charge of -2. Right next to that, you've got fluorine. It only needs 1, so it will form an ion with a charge of -1. What about helium? Helium has 2, right? They're both in the first orbit, right? Is helium going to want to share or take away electrons or give up electrons? Helium and anything in that last column will never react chemically. Make sure you get that down. They will never ever react with anything chemically. They're called the inert gases because they are inert.
What are they called?
Inert, I-N-E-R-T. You may find some older references call them the noble gases. I don't know why.
I've heard that.
And those will not have a chemical reaction.
They will never, ever combine with another chemical to form something. So there are 3 types of chemical bonds. The strongest are called covalent. Covalent. Those are formed when electrons are shared between atoms. Generally, that's going to be anything in the same column as carbon or to the right of it. The same column as carbon and to the right. Hydrogen is kind of like carbon in that it's unique. Most of the time it acts like a +1, but it can also gain electrons and act as a -1. Okay, so when you see H2O, that's 2 hydrogens and 1 oxygen, right? They are covalently bound. Make sure you get that. Covalent bonds are the strongest. Everybody got that? The second kind are called ionic bonds. Ionic bonds. These are not as strong as covalent bonds. They are formed between ions with the opposite charge, ions that have an opposite charge. So if you take a look over there in the first column and you scroll down, you'll probably see Na. That's the symbol for sodium. Sodium. If you look over in the next to last column, you might see Cl for chlorine. Everybody found those? Since sodium is in the first column, it's going to be a +1. Since chlorine is in the next to last column, it's going to be a -1. +1, -1, they're going to cancel each other out, aren't they? Sodium is a metal that is so reactive, you cannot store it in an open container. You have to cover it with oil or it is going to react with the oxygen in the air. If you take a chunk of it, about an inch square, and you throw it into, say, a toilet bowl, it will explode with enough force that it would rupture that. So it's quite reactive. Chlorine, I think you all know that chlorine was used as a chemical weapon in World War I. I actually spent 8 weeks one summer working in a chlorine plant. They had about 2 million pounds of the stuff stored on site. You had to have a respirator with you everywhere you went. They just give you this little box that has a tube coming out of it. And when I say everywhere, I mean everywhere. You go into the bathroom, over the urinal, there's a shelf for you to put your respirator on. Why? Because it is so reactive. It literally would burn your lungs if you breathe it. So those sound like two pretty bad news things, don't they? But if you combine them with an ionic bond, does anybody know what we call that? Salt. Salt. Sodium chloride. Probably sprinkle it on your food every day. So I hope you get the idea that chemical bonding changes a lot of things, doesn't it?
So, like, how does it change it to, like, to something that can kill you to something that's not as like doesn't harm you at all?
Because remember, sodium wants to kick out an electron, right? Chlorine wants to grab an electron. If you breathe in pure chlorine gas, where is it going to find the electrons? In your cells, right? Everybody see how that works? But once they have that full shell or that full orbit, they're non-reactive. Sodium is giving up electron, isn't it? Chlorine is right there to receive it. That make sense? It's a good question.
If we want to print out the periodic table to like write on it and stuff, is there a specific one you want us to find?
You
or-
can choose any one you want. Every single classroom I have ever been in, science classroom, up until the last 10 years, had a periodic table hanging somewhere on the wall. And I always figured if it's on a poster on the wall, it's not a cheating aid. It's a study aid. We have moved on from paper. I know every single one of you happens to have some kind of internet connected device on you at this moment. So if you wanted to look up a periodic table from the parking lot of Wendy's, you could. So I'll let you have one for the exam. Does that sound fair? I will ask to see it, but I'm not overly worried about anybody cheating because, like I said, I think it's a fair tool. Okay, good question, by the way. Any other questions? Last type of bond is a hydrogen bond. Hydrogen bond. We already talked about those on Tuesday. Remember those temporary bonds that form between water molecules? So the strongest are covalent, middle strength would be ionic, and weakest would be hydrogen. And make no mistake, ionic bonds are not very strong. They are considered permanent, where hydrogen bonds aren't. But you can break a ionic bond simply by stirring. You put water in salt and water, stir it, it's gone, right? So anybody have any questions about the three types of chemical bonds? That would be a great list and describe question for the exam.
What is hydrogen bond? Like, why is it the weakest?
Because it's temporary. It only forms between polar molecules. You remember how water was bent like this? It had a slight negative pole and a slight positive pole. Hydrogen bonds are interesting because they're not considered permanent. They're fairly weak. Well, not fairly weak, they're very weak. But if you get enough of them, it really makes a difference in how the formula behaves or how the substance behaves. That's why water is such a good, remember we talked about being a good heat sink? That's because it's got more hydrogen bonds than we can count, really. You started to say something?
I was gonna say, like, would a hydrogen bond, like, would an example be like water and salt water, and like if you heat it up, they just
Yeah.
yeah.
Any other questions? Last thing I want you to know, the electrons in the outer orbit are called valence electrons. Valence electrons.
Yeah, sorry.
Yeah, the outer. And that's called the valence orbit. The valence orbit. I don't know about you, but I think at this point I'm okay with saying that's it for the chemistry. Does that sound okay to you all? It's enough of a background that you can see where things go, how they fit together. It's enough for us to do experiments and have an understanding, but I don't think we need to go any deeper than that because this is not a chemistry course. So everybody's cool with that?
And the chemistry, like, there's gonna be like parts of it on the exam.
Yes. Just know what the atomic number is, atomic mass. Know the difference between an ion and an isotope. Did we talk about isotopes? Okay. An ion is a difference in charge, right? It's where you have a different number of protons and electrons. You either have more electrons or fewer electrons. What happens if you add more neutrons? Neutrons, by definition, are neutral, right? The only thing they add or subtract is mass. And mass is just a fancy term for weight. Take a look, if you will, at hydrogen, upper left-hand corner. Atomic number is 1. So how many protons does it have? But notice the atomic mass is also 1. It does not have any neutrons. Is everybody clear on that? It does not have any neutrons.
So an isotope has no neutrons.
Huh?
Isotope has no neutrons, right?
No, I'm getting there.
Oh.
However, there is a form of heavy hydrogen called deuterium. Deuterium. D-E-U-T-E-R-I-U-M. Deuterium. Deuterium has an atomic mass of 2. So if we take away the one proton, what have we added to it? A neutron. Does everybody see that? We know that hydrogen gas is explosive, right? If we filled a balloon with pure deuterium and struck a match, what do you think it's going to do? Because it's still hydrogen, it's just heavier. An isotope is an atom with a different atomic mass. It is either heavier or it is lighter. It is either heavier or lighter.
Has a different one.
Atomic mass. In other words, it's got more neutrons or less neutrons. Is everybody clear on that? Chemically, all isotopes behave the same. Chemically, all isotopes behave the same. Here's you a fun little question. True or false? An atomic reactor can blow up in the same fashion as an atomic bomb. How many of you think that's true? How many of you think it's false? How many of you are unclear on the concept of democracy in action and refuse to believe in the power of the vote? That might explain something. Here is your truth. There is absolutely no way possible for an atomic reactor to explode in the fashion of an atomic bomb.
It would, it wouldn't blow up as an atomic bomb. It would make just like
Meltdown.
basically meltdown and expel a lot of gas.
Yeah, but it still is not exploding. The reason for that is two uranium isotopes. There's uranium-238, which is the one that you find common in nature. It's radioactive, but it's generally stable. It has a half-life of billions of years. Uranium-235, which has 3 less neutrons, is explosive. Y'all heard of Oak Ridge, Tennessee, the atomic city? Just down I-75, about 20 miles east. It's home to the Manhattan Project. It's where they built the first bomb that they dropped on Hiroshima. It's also home to the United States Nuclear Uranium Depository. It's where they keep all of the bomb-grade uranium. You may hear bomb grade or weapons grade, and that is highly purified uranium-235. Uranium-238 can never ever explode. What they use in a reactor, they call it reactor grade or fuel grade, and at most it's going to have about 30% uranium-235. Pretty wild, isn't it?
That's the one they use in power plants, right? The ones that are stable. Okay.
So if it, say that had a meltdown, it would still do a lot of damage, right? Because it expels like
Meltdown literally means exactly what it says. If you have an uncontrolled reaction going on inside a nuclear reactor, it's going to get so hot it literally melts the uranium. Uranium's a metal. Then it's going to melt the container. Is the coolant still going to be circulating? Then it's going to melt the concrete below it, and it's going to melt the dirt, and it's going to sit there forming a big slag heap like at Chernobyl.
it'd be an elephant foot.
Yeah. How'd y'all let me get off on these rabbit trails?
Hey, it's fun.
Well, you learn something new every day, don't you? But I want you to understand, isotopes chemically are no different. They react the same. Your body cannot tell the difference between carbon-12 or carbon-14, for example. We'll talk more about those when we get to things like dating. You've heard of carbon dating, radiometric dating. They use radioactive substances. Most isotopes are radioactive. Different isotopes are generally radioactive. They're unstable. So that's it for the basic garden variety chemistry. What we're going to talk about next are molecules. There are a lot of molecules that are important to life. We got into a fairly in-depth discussion of water on day one. You're going to see some like ATP that are used for energy. Water, ATP, salt, small molecules, 2 or 3 atoms, nothing too exciting, right? For life to exist, you're going to need something called macromolecules. Macromolecules. What's a macromolecule? What does macro mean? It means big. These guys are huge compared to something like water. There are going to be 4 different classes of them. For each one, I want you to know the basic unit, the type of bond, the type of chemical bond, the primary function for living things, the primary function, and then 1 secondary function. I think that's a reasonable amount, don't you? 4 things and 4 classes. Sound okay? So, the first one we're going to talk about are carbohydrates, carbs. The basic unit of a carbohydrate is called a monosaccharide. Monosaccharide literally means 1 sugar. I'm sure all of you have heard of glucose, haven't you? Glucose is a very simple sugar. Do what?
How do you spell that?
MONOSACCHARIDE. Monosaccharide. Table sugar, sucrose. It's a disaccharide. It only has 2 sugar molecules together. You see how this works? The main function, the chemical bond is called a glycosidic bond. GLYCOSIDIC, glycosidic. How many of these monosaccharides can you put together? Pretty much as many as you need. They can get up into the millions. Who should I pick on? All the way there in the back, what's your name again?
Deanna.
Huh?
Deanna.
Deanna. What would you think is the main function of a carbohydrate? Can you help her? I can tell you all are not at the age yet where you have to worry about things like diabetes, which is a good thing. Y'all keep eating sugar the way you are, you probably will end up like I am and be a diabetic. Short-term energy storage. Short-term. And underline short-term because this is not something that you're going to be packing around for a long period of time. If you're out there running a marathon and at the halfway point, which is 13.1 miles, you're given something to eat for quick energy, you're not going to ask for a Baconator from Wendy's, are you? You're going to ask for something that's got some carbs. So short-term energy storage, it breaks it down very quickly. You need an energy boost, get you some sugar. The secondary function, I'm gonna just say structure. Structure. You're gonna find that structure is the secondary function on pretty much all of these. Although you can get some weird types of structure. Anybody here ever heard of cellulose? You ever heard of fiber or roughage in your diet? Basically, cellulose is what they make paper from. It's those fibers that you find in leaves, grass, so forth. It's not the hard part of wood, it's just the fiber. Chemically, it's nothing but a chain of glucose molecules. But your enzymes can't recognize it, so your enzymes cannot break it down, so it passes right through you and helps clean out your gut. That's why you have to get so much fiber in your diet. It helps move things along. If you get too much, you get the rest. It's just sugar, though. We just can't digest it. Y'all ever heard of chitin? C-H-I-T-I-N. You may want to get that one done. You ever stepped on a bug or a beetle? Makes that nice, satisfying crunch. That's because the exoskeleton of that beetle is made up of another structural carb. It's called chitin.
Can you spell that again?
C-H-I-T-I-N.
Just a little fun fact, because I learned this the other day. Mushrooms are actually, a lot of mushrooms have a layer of chitin, and that's why you need to cook them down, because it cooks down the layer.
They sure do. Their cell wall, I think that's actually the main component. Good job. And people ask me why I like biology. There's all kinds of fun stuff out there. So everybody got that?
I have a question.
Yeah.
You teach 4 classes, right?
Yeah.
Yeah. Like that, which one's your favorite out of all of them? You did pick.
Probably this one, because here I get to talk about all of biology, not just the human body. Anatomy. It's fun, it's interesting, but after a while, there's only so many ways you can describe the skeleton. Some of those names for some of those muscles, makes me glad Latin is a dead language, because I wouldn't speak it otherwise. We talk about anything and everything. We'll talk about biological warfare, we'll talk about origin of life, we'll talk about evolution, we'll talk about genetics, gene therapy, genetic enhancement. Yeah, there's a lot of good stuff out there. Controversial stuff.
Question, where's the bathroom at?
Go down about halfway down the main hall until you come to a 4-way intersection.
Okay.
Hook a right, second door on the left.
Okay.
Make sure it's the second door, the first one's the window.
Thank you.
Learned that the hard way. So, where were we?
Chitin.
Chitin, okay. I'm starting to go back to nuclear bombs. Any questions about carbs? Bottom line is, we use them for short-term energy storage. You may have heard of starch in plants. You ever heard of that? Potatoes, a prime example. Those are just long chains of glucose. It's one of the reasons why they tell you to eat something like potato chips in limited fashion if you are diabetic or pre-diabetic. And me is like, didn't I see him drinking a Dr. Pepper? She did. I'm allowed to cheat once a day. And I needed something on Tuesday. So everybody with me? All right, next up, we've got lipids, L-I-P-I-D-S, lipids. Their main unit is a fatty acid, The type a bond is an ester bond, E-S-T-E-R.
Ester what?
Ester, E-S-T-E-R, that's the bond.
What's an ester bond?
So what on earth are lipids? Well, the non-scientific term for them would be fats, oils, and waxes. Fats, oils, and waxes. Basically, these are nothing more than long hydrocarbon chains, long hydrocarbon chains. Carbon bound to carbon, bound to carbon, bound to carbon, hydrogens all along the side. Who can tell me what their main use is? I'm just trying to learn names here, so I have to pick on everybody. Young lady in front of Mr. Sparks here.
Reagan?
Huh?
You just asked my name's Reagan.
Reagan. What do you think is the main function of a fat? It's okay if you don't know. I'm just saying, you want to call on somebody, pick a friend? No one? Long-term energy storage. Long-term. You get more bang for your buck by breaking down a fat molecule than you do by breaking down a carb. The only problem is it takes a lot more effort. It takes a lot of time. If you eat a lot of carbs, you have an excess amount of calories, don't you?
So would be protein be long-term storage?
Protein won't even register on the radar screen. You're jumping ahead, sir. I love the enthusiasm, but we gotta walk before we can run.
I had to guess at least.
It's a good guess. That's one of the secondaries of protein. What's the difference between a fat and an oil?
Solid and liquid?
Huh?
Solid and liquid?
At room temperature, an oil is a liquid, a fat is a solid, and that's the only difference. Like I said, real technical, isn't it?
So like cooking oil is a long-term storage, I'm guessing?
The secondary use is interesting. First up is insulation. Insulation. You ever look at people who live in the Arctic and they tended to be fairly stout, much shorter. Less surface area, they probably have a little bit more fat because that helps them retain their heat. Whales, you've heard the term blubber, it's literally whale fat.
Did
It
they use
used
it?
to be that they would harvest whales solely to get the fat so they would have fuel for oil lamps. Gives you an idea how calorie rich it is, doesn't it?
And then the one whale went out and killed sailors, and that became the book of Moby Dick.
Yep. The Essex. Interesting real life twist there. The other secondary effect is where waxes come in. A wax is just an extremely hydrophobic lipid. It is an extremely hydrophobic lipid. Hydro meaning water, phobia meaning fearing. In other words, waterproofing. Waterproofing. Waxes are great waterproofers. There are certain plants that they basically have the surface of the leaf coated in wax so they don't lose water. Those are your main sources of energy from your diet. Is everybody clear on that? Which brings us to our third one, proteins. Huh?
Yeah, anyway.
Proteins are made up of amino acids, AMINO, amino acids. The type of bond is called a peptide bond. A peptide bond. The primary function of a protein is catalysis. 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 A catalyst. The proper term for these protein-based catalysts is enzyme. 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 Life is not possible without enzymes. Period. Bottom line. End of story. No proteins, no enzymes. No enzymes, no life. I cannot emphasize how important that is. What do enzymes do? They lower the energy of activation of a chemical reaction. They lower the energy of activation. Could I burn this?
Yeah.
Can I make it ignite just by holding it? No matter how high my body temperature is, it's not going to be enough to ignite that, is it? What's your name again? Lillian. What would I have to do to get this to ignite?
Set it on fire.
Set it on fire. Need to strike a match or a lighter, wouldn't I? Is there another way I could?
By the sun.
Okay. If I had a magnifying glass, I could focus sunlight on it. Good job. I could set it on something that's much hotter than my hand, right? The burner on a stove. You get an idea. All three of those require a fairly high amount of energy. That energy is called the energy of activation. In other words, we don't get energy unless we spend energy. Any questions on that? Has anybody here ever made caramel? To do it, you basically take table sugar and you heat it up in a pan on your stove until it turns nice golden brown and gooey. You literally are melting it. Would any of you like to try some homemade caramel? Would you be willing to stick your finger in the hot sticky goop while it's on the stove? So how is it that you can actually burn sugar for energy? Your answer are enzymes. Rather than having to heat the sugar up to that level of warmth where it would damage your tissue, instead, your body brings those molecules together and breaks them down. Does everybody understand how enzymes work now? Everybody stop. Stand up. Stretch if you wish. Okay, we're done. I just want to give you a chance to move. Stretch if you want to. You're starting to get that 1,000-yard stare. Don't feel bad. I hate talking about biochemistry. Biochemistry nearly broke me. So how many pages of notes you got?
Count Tuesday or just today?
Just today.
Probably
4 or 5. Oh my
3.5.
goodness.
I don't know. Yeah.
Well, we're definitely running a little bit longer than we should. This is what happens when you all don't talk, but I do, see? All right, proteins have a variety of secondary functions. The first is energy storage. The second is structural. Third, transport. They actually help move things around, ways that you might not realize. What makes proteins such a versatile molecule is their structure. They have 4 levels of structure, and unfortunately, we're going to need to know all of them. Primary structure of a protein is simply the sequence of amino acids in the chain. The sequence of amino acids. So alanine, alanine, threonine, alanine, alanine, phenylalanine, you get the idea.
Sequence of what?
Huh?
Sequence of what? Sequence.
Of amino acids. That helps determine the basic chemistry of it. The secondary structure is how it arranges itself in 2 dimensions. So let's pretend that my power cord here is an amino acid chain. Okay? I'm holding it flat and getting kind of a couple of loops. Everybody see that? I can pull them together tighter, can't I? I can twist it around on the side. All of those are secondary structure. We're basically manipulating it in 2 planes. Front and back, side to side. We're not doing it in the 3rd plane. Is everybody clear on that? Next up is the tertiary structure. T-E-R-T-I-A-R-Y, tertiary.
You go Bissell for that?
Huh?
You go Bissell for that?
T-E-R-T-I-A-R-Y. Primary means first, secondary means second, tertiary means, not surprisingly, third. Put a star by it. This is by far the most important one. And it is the 3-dimensional folding of the protein, the 3-dimensional protein. Everybody see how I folded that in 3 dimensions? It's almost like I created a little basket in there, isn't there? Everybody see that? And that's why it is so important, because it literally does create pockets, baskets. That's where things can bind. If you alter the tertiary structure, you will probably destroy the protein. If you alter the tertiary structure, you will destroy the protein. The easiest example of that I can think of frying an egg, something that we've probably all done or seen done, right? When you crack an egg open, you get 2 parts, don't you? You get the yellow yolk, which is more or less a solid, would you agree? It's kind of goofy or gushy, squishy, but it's still a solid, right? The part we call the egg white is an actual liquid, isn't it? It's not white when it comes out of the egg, is it? It's more or less transparent. It's quite runny, isn't it? It only becomes a solid white thing when it hits that hot oil. The reason for that, this is a group of proteins called albumins, and they are soluble in water. It hits that hot grease, it unfolds the protein, it destroys the tertiary structure, and it now is a solid. You cannot unfry an egg, can you? See what I mean when I say you kill the 3D protein, or 3D structure, you kill the protein.
So like
Period.
a marshmallow, like once you fry it, it's just
A marshmallow, I would say, is probably all carbohydrates. Believe it or not, sugars are sticky. And some of the old movies, like 40s, 50s, 60s, when they would do a chase scene, they would actually prepare the road for the vehicles. And you know how they would do that? Big barrels of Coca-Cola. They would actually roll the barrels of Coke down the street, let it dry a little bit and get nice and sticky and tacky. And I think we've all had soft melted marshmallows. They stick on everything, don't they? Don't ever take a child to a place where they're having a marshmallow roast. They will create a mess. You will never ever see any of it. I hate to say it, but snot. Again, if you've ever been around children, you know that snot is sticky, right? Well, it actually plays a role in defending your body. Did you know that? Anything that you breathe in that you don't want in your lungs, hopefully it'll get caught up in that. Whenever you have a respiratory infection, your nose gets really runny. That's because your body is essentially putting way more liquid than sugar into the mucus to try to flush the invader out. Pretty wild, isn't it? Told you you'd learn something in here. Now, how did I get on this stickies? You're a bad influence on you. You brought up marshmallows. I brought up fried eggs, bacon, and now I'm hungry. I'll be stopping by Wendy's on the way home. Okay, so back to proteins. There is a fourth level called the quaternary. 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 That's the fourth level. We're going to call this the assembly of subunits. The assembly of subunits.
What was the last word again?
Assembly of subunits.
Can you spell it?
For example, I'm sure all of you have heard of the protein hemoglobin, yes? Hemoglobin transports what, Mia? Oxygen. It's the main protein in your blood, right? What you may not realize, it's actually made up of 4 separate peptide chains. 2 alpha and 2 beta. You have to put all 4 of them together to have a functional heme group. Well, let's not worry about it any more than that. Is that okay for everybody? We don't need to get bogged down in biochemistry. And this brings us to our last macromolecule. Well, last class of them. Nucleic acids. basic unit is called a nucleotide. A nucleotide. Phosphate bonds hold it together.
What holds them together?
Hmm?
How does it?
A nucleotide and phosphate bonds. Now, when it comes to function, these don't have a nice clear-cut thing. They're come in two flavors, DNA and RNA. DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. No, you don't have to know that or write it down. DNA has only a single function. Information storage. RNA, the supermolecule that does everything. Literally. You want to use it for structure? Use it for structure. You want to break it down and get some energy from it? Break it down. You want to move things around, use it for transportation? Go for it. You want to use it as a catalyst like an enzyme? Believe it or not, there are some RNA molecules that act as enzymes. We call them a ribozyme, R-I-B-O-Z-Y-M-E. And there are even certain viruses that use it to store genetic information. Retroviruses. HIV, you've heard of that one, right? It's a retrovirus. It uses RNA. RNA is the exact opposite of DNA. It's the Swiss Army knife of biology. It does everything. But it has one teensy tiny little problem. It is the most unstable molecule there is, in my opinion. And yes, I'm speaking out of bitter experience. I tried RNA work one time. Here's what I had to do. I had to order brand new ceramic and glassware that had never been used before. I then had to wash it in hydrochloric acid. I then had to dry it in an oven for 2 weeks to make sure it was completely free. And then I had 1 hour to go all the way from a freshly plucked leaf to RNA. And that hour had to be spent in a walk-in freezer in the middle of January. Sound like a lot of effort? It was. Did I get any RNA out? No. It broke down, even despite all that. My advisor laughed at me and said that happens to everybody on their first try. Do it again. I laughed at him, said, good joke. He said, I'm serious. I handed my keys and said I quit.
Where was this at?
Huh?
Where was this at?
Vanderbilt. He realized I was also serious. That was the last time either of us discussed quitting or RNA. It's so versatile because it can do so many things, but its very instability is what allows it to. Contrast that with DNA. DNA does only one thing. it stores information. But let me tell you, it is the most stable thing in existence. You want to hear time for one story? You like stories? Anybody here ever heard of Masada? Masada is a mesa that's found in the Judean desert. After the Romans attacked, sacked, and burned Jerusalem, about 1,000 men, women, and children fled to Masada because 75 years before this, King Herod had built a palace up there and a storeroom that was chock full of food. It's out in a desert that gets less than an inch of rain a year. They had big cisterns carved into the rock beneath the mountaintop, so they had all the water they wanted. Romans being Romans, they can't let any group go unpunished, right? So the Romans then proceeded to build a 250-foot tall ramp with a 3-story siege tower that they winched to the very top of the mountain, put a battering ram on it, and started banging down the walls. The Jewish zealots grabbed some of the wooden beams from King Herod's throne room and built an inner wall made of wood that couldn't get knocked down. The Romans set the wood on fire. The wind shifted back and started melting the head of the battering ram. Night fell. If the wind stays this way, the Romans' tower burns. If it flows back, then the Romans will have burned the wall. The wind shifted during the night. The Romans stormed it the next morning to the sound of silence. Almost 1,000 men, women, and children had committed suicide rather than submit to Roman rule. In the process, a basket of dates, y'all know the fruit of the palm tree? A basket of dates was in a storage room that collapsed, had the wall and the roof fall on it. The storage room caught fire and burned, and 2,000 years later, when they were digging up Masada, they found the basket of dates, including the seeds. Pretty cool. Used one of them to radiocarbon date the thing, set it up on a shelf in a lab in Israel for 30 years. 30 years later, somebody gets the idea, I wonder if we could plant those seeds. Now think about how long they'd sat there. I don't know about you, but I wouldn't want to eat 75-year-old food from World War II and it's been stored in a freezer. We're talking an open basket sitting out there 75 years in the desert. Then has a roof fall on it, it burns, and is buried for 2,000 years. Guess what happened? One of them sprouted. That's how tough DNA is. See ya. If you're in lab, we're in room 135, down the hall on the left. Would you like to be the key master? Like that story?
Yeah.